Next, uh, good morning once again. Now we have one important speaker from Malaysia, Dr. Pasupuleti Visweswara from Universiti Malaysia, Sabah. And he is going to deliver a talk on medicinal plant mediated biosynthesis of nanoparticles for human diseases. I am happy to share something about him. He is our alumni. So I request him to start his presentation now.
Yeah, thank you so much, sir, for having me here with this. Thank you so much, sir, for having me here in SLI International webinar on role of nanotechnology in current scenario. My sincere thanks to Professor Sudarshanam, sir, for having me here with this webinar, and also all the organizing members and my co-speakers as well. Okay, the organizing committee, including the Vice Chancellor, Rector, Registrar, Principal, and also Head of the Department of Botany. My sincere thanks to you all for having me here. Well, today I'm going to talk about medicinal plants mediated by biosynthesis of nanoparticles for human diseases. And before going into nanoparticles or medicinal plants, but we must understand what is the importance of health and why every time even our seniors or elders or ancestors say health is wealth. So if you have health, so you have everything with you. So which means once you have health with you, so you can achieve anything, you can earn any kind of wealth, but you have wealth. Now you can observe the pandemic situation in India and across the globe as well, how a single, invisible virus is killing the human beings. So even though we have lots of money, we cannot do anything. So that's the reason health is most important. And the main focus of my talk would be on antioxidants and oxidative stress, and also biological synthesis of nanoparticles. So here, coming to next one, the health only, whatever the information I'm providing here, at the end of the day or at the end of the presentation, it will be completely linking with the health and human wellness. So nano, when we talk about nano or nanotechnology or nanosciences, it is not actually a very new word, or it is not a very new subject suddenly appearing, from nowhere. No. Since many years, since few decades, nanotechnology and nanoscience exists in the nature. Even many of the scientists from developed world, they are doing research on nano. Nano means it's very small, which we cannot see with naked eyes. And usually the particles of nano range, be between 1 to 100 nanometers of range. And here you can see different materials which I am showing, like water molecule, glucose molecule, antibodies, viruses, bacteria, cancer cells, and a period, tennis ball, whatever you take, they are composed of so many atoms, so many molecules, and each molecule will have its own diameter. Whenever you talk about diameters or nanometers or millimeters, so it's a bit huge amount when we compare with nanotechnology or nanoparticles, because the simple basic principle is that when the size is being reduced of a molecule or an atom, the surface area volume, we call it as surface area volume, will be very much larger. The more or the smaller the molecule is, the more the surface area is. So when you have more surface area, you have more absorption, whatever it may be. It can be a drug, or it can be a plant material, or it can be emulsions, it can be nanoformulations, whatever it may be. The absorption of these nanoparticles will be very high, very huge. So that's the reason n number of areas of expertise or n number of research areas or departments. They are using this nanotechnology in every field. Say, for example, engineering, yes, and basic sciences, yes, pharmaceutical, yes, biotechnology, cosmeceutical technology, nutraceutics. So where not? You can name whatever the field exists in the nature, they are using nanotechnologies. And as I mentioned just now, nanotechnology, basically the particles range between 1 to 100 nanometers with very minute size, which we cannot see with the naked eye. And also it has got higher surface volume or larger surface volume. But if you observe, as I said just now, the nanotechnology is not very new. It is existing since few decades. See, in 1959, Richard Feynman, he has already introduced nanotechnology word. Okay, the concepts of nanotechnology. But definition was given by a Japanese scientist later on in the 1960s. So that is why we are now talking about the old picture in a new gallery. So this is actually seems new proverb, which I tried, and I was thinking, old wine in a new bottle. So I was just thinking, and then I have come up with this particular quote, old picture in a new gallery. So, which means we are talking about the old matters, but again, it's so fresh. That's why if some of the continents, say for example, like in India, we usually say old is gold. So now we are talking about old technologies, and we are talking about old applications, the old technologies, old applications, and now even more new things we can find. So that's what I was mentioning, 1960s and 70s, 50s, the nanotechnology was evolving. And in 74, Norio Taniguchi, a Japanese professor, he has defined what is nano and how exactly nano looks like and what exactly happens when a nanoparticle or nanomaterial formed. Because it all deals with the diameters, it all deals with the range of the particle size, because as I have mentioned just now, the particle size, the lesser the particle size, the more absorption will be there. So here you can observe it consists of processing of separation, consolidation, and deformation of materials by one atom or by one molecule. It usually happens not in one step, straight away, just like that. It has several steps. That is why we say it as separation. They have to be separated, then consolidation, again deformation. That's how usually nanotechnology or nanoparticles or nanoscience is being educated everywhere. So this is what we usually follow when we synthesize the nanoparticles using biological systems or using physical systems or using chemical systems. Whenever we talk about nanoparticles, the main thing comes to any researcher's mind is the size. And next one is stability. Next one is applications. What exact application it's going to be used. So these are three different types of modes or methods we can synthesize the nanoparticles: physical method, chemical method, and biological method. There are three different varieties or three different methods in physical mode, which is evaporation, condensation, and also laser ablation. If you take in chemical, it's Turkish method and also BSS, we call it as burst reference synthesis. But even though we have physical and chemical methods, they are using lots of physical methods and chemicals are involved in that. That's why my main interest to explain or to mechanistically evolve or mechanistically delve into the things through biological systems, using biological sources as the primary resources for the synthesis. So even here, if you observe biological synthesis, they have microbial systems, fungal systems, algal systems, and plant systems. But Even though microorganisms also are the source material for synthesizing the nanoparticles. Here, in all my research throughout my career, more than a decade, excuse me, I am using plant materials as the source materials to synthesize biological nanoparticles. So that's why we call them as biological synthesis or biosynthesis, because biological molecules are being used as the source material and they will be combining with the metallic ions. Finally, lots of processes will go on once we combine them together. Then finally, we get the nanoparticles, which are considered as safe when compared to the synthetically or chemically produced nanoparticles or drugs. So when we talk about mechanism, or when we talk about formation of nanoparticles or synthesis of nanoparticles, we have different modes actually. One is top-down approach, another one is bottom-up approach. If you observe on your right, top-down approach, which means a bigger macromolecule becomes micromolecule, micromolecule becomes nanomolecule. So that's how usually it happens, which means we take a big rock and making into pieces, finally it becomes a nanoparticle. So that's how macro to micro to nano. And bottom-up approach, so this is usually piling. So we take the building blocks, which means atom by atom. So thereby we try to synthesize the nanoparticles. These two approaches are acceptable, but based on the source materials and based on the size of the source materials and also the metals or metal ions. If you observe the synthesis or the mechanism, how exactly it happens. So you observe here, you can see the metallic ions, which are in purple color. So the metallic ions, then these are the bioreducing agents. In our case, the plant materials or the plant molecules are the bioreducing agents, which means biological reducing agents. So they have lots of other, functional groups, whether it can be OH, it can be CHO, it can be anything else. So they have the functional groups which has the capacity to function as the antioxidants or to give more biological activities. Say, for example, antioxidant, antimicrobial, anti-cancer, anti-diabetic, and so on. So once we merge them together, the reduction happens. Once the reduction occurs, then it is being reduced like reduced silver atoms. Once the reduced silver atoms, it grows, finally it has a collision or colloid kind of thing. They appear or they formed as nanoparticles. Here I have mentioned gold or silver, but there are various types of metals are being used to synthesize the nanoparticles using biological sources as the reducing agents. And finally, we check the stabilization. As we all know, medicinal plants usually, play a very important role in various types of diseases. And also not only that, even lots of herbs, we humans in Asian countries, we take raw herbs as food or as supplement or as medicine. So herbal medicines or herbal formulations, or India is very much known for Ayurveda, and the other countries are also using, Ayurveda, Chinese traditional medicine, we call it as TCM, CTM. So all these combined together, we call them as traditional or complementary medicine. So these traditional complementary medicine always have good benefits to the human beings, and also they usually do not have or do not possess severe side effects. They are with very minimal side effects or no side effects for sure. And that's the main reason why researchers always try to use medicinal plants or natural products or herbal medicines or herbs to synthesize or to prepare the crude extracts, or to prepare the isolated forms, or even prepare the nano formulations. So it is easy to treat the people with these kind of formulations. As I was mentioning, nanotechnology and its applications have widely been entered into various types of fields, which you can see food and agriculture. Actually, in food packaging and agriculture, they are using very vigorously this nanotechnology and nanoparticle research products. And energy environment, even energy, solar systems, and other environmental areas, they are using nanoparticles very regularly. Biotechnology, of course, even diagnosing the diseases, even industrial biotechnology also, they are using this nano science and nanotechnology applications. Herbal medicine, definitely, most of the herbal formulations we mix with metallic ions and thereby forming the or synthesizing the nanoparticles, and we try to give into the rats, or sometimes we can give into the animal models usually. So in vivo, then we test them. And finally, some of the nano products already out in the market for human usage, especially like cosmetics, nutraceutics, and also some supplements. Textiles, also they are using. Medicine and health, definitely for drug targeted therapies, and also prompt therapies, site specific therapies, etc. And aerospace, yes, for the lightweight materials they are using in aerospace, robotics as well. Information technology, yes. If you observe the size of the laptop, size of the mobile phone, size of the electronic gadgets and other stuff, when compared to the, say for example, compared to a decade before, so now the size of the materials are very less and very light. This is also due to nanotechnology and their applications. So these are three different points I have given for the viewers, easy understanding. They are the fundamental building blocks of nanotechnology, and its starting point for preparing nanostructured materials and devices. And nanoparticles are also useful for various kinds of researches and various types of researches as well. Because as I mentioned earlier, it's mainly because of large surface volume. Even this one I have mentioned, but here I wish to inform you that there are various types of metallic nanoparticles are being synthesized by researchers or scientists. So silver nanoparticles being prepared using silver nitrate, selenium nanoparticles, gold nanoparticles, copper nanoparticles, even zinc oxide nanoparticles, so titanium oxide nanoparticles, various n number of metals are being used to synthesize biologically synthesized nanoparticles. And here we need to remember one thing is the nanoparticles can be synthesized using single metal ion or single metal salt or combination of metallic salts. So it depends on the requirements, it depends on the objectives, it depends on the compatibility of the synthesis. So probably there are some reports saying that some of the researchers they use the combination dual metal nanoparticles as well. So this is how I usually do. So we use various types of plant materials so far, and then we mix with metallic salts, formation of nanoparticles. While formation, there is, as I have mentioned earlier also, there will be reduction first, then again capping, then stability. So these are the various processes and steps happen when the nanoparticles are being formed using biological source materials. So here in this picture, you can observe the yellow color and the purple color or dark color. This is actually the formulation. So the formation of nanoparticles with a color change. This is actually a layman signal that we can confirm that nanoparticles are being formed. And second thing is, to scientifically prove it, we cannot just say that, okay, this has been changed from this color to this color, so nanoparticles are formed, so this is the nanoparticle. No. So to prove it scientifically, we have to go further for the characterizations of biologically synthesized nanoparticles. So then we have confirmed with UV spectroscopy and XRD to confirm, this is to confirm the formation. XRD is to confirm the shape and also the structures of the molecules or particles. This is FTIR, whereby we try to use this FTIR to know which functional groups are really active in in the synthesis of nanoparticles. And if you observe this picture, this is actually electronic microscopy, which is SEM or TEM usually we use to identify or to confirm the size of the different particles available in your particular solution. And last one is here, you can see the graph here. So this is actually particle analyzer. This is 100% shows, this usually shows you the, or gives you the exact size of the particles. Once you finalize this characterization, we will go to the applications. It is again based on the experimental design you have. So we usually do antimicrobial activity, anti-diabetic, antioxidant, and even anti-cancer. So this is a hypothetical diagram which I have been given in one of my articles, and this one is anti-cancer activity, so which I will be showing in the next slides. But basically, when we talk about nanoparticles and biological systems or human health or disease, all of us must remember that oxidative stress is the culprit of all sorts of diseases. Because when we have stress, say for example, in a layman language, when we have stress, we say, oh, I am tired. But when the cells have stress, we call it as oxidative stress. Oxidative stress is the one which is actually an imbalance between reactive oxygen species or free radicals and antioxidants, which are the defensive systems or defensive mechanisms. So when there is an imbalance between antioxidant system and free radical system, then the cells or tissues feel oxidatively stressed, so much stress. So that is why to make the cells healthy, to keep the tissues healthy, and to keep ourselves healthy, the free radicals, number of free radicals or the quantity of free radicals must be reduced. So antioxidants usually does that work. So this is another pictorial representation of pro-oxidants, which are not good for health. When they are high, then we call it as oxidative stress. So antioxidants are actually less in the body when these pro-oxidants or reactive oxygen species or reactive nitrogen species, or commonly we can call them as free radicals, which are always with the free electron vacancy. So that's how these free radicals or these ROS, RNS will harm the tissues or cells, whereas antioxidants always protect our cells. This is the equilibrium when you have antioxidants and ROS, so no harm. If you have ROS excessive, then you have oxidative stress. You have more ROS, you have excessive oxidative stress also. But when you have continuously excessive oxidative stress within the cell or tissue, then that's going to be harming the cells and also tissue, finally the organ as well. So nanoparticles and their biological activities, what I have reported so far. So I have reported in 2013 in one of the premier journals using these nanoparticles, synthesizing the nanoparticles using one of the plants. This is also from Tirumala Hills. And then we have proceeded with antimicrobial activities on different microorganisms. So we have clearly reported that this particular plant material and biological synthesis of nanoparticles are really good to inhibit these microorganisms. However, this is another article written by me, and I have given 3 different mechanism of actions based on my understandings through my research, and also reading lots of other information as well. So I came to a conclusion, and I have given these 3 different mechanisms, how the nanoparticles interact with bacteria, and then they can finally kill the bacteria. First method, if you observe, it is interaction of nanoparticles with enzymes. Finally, they inhibit the signal transduction. Once they inhibit the signal transduction, finally cell lysis occurs. So when cell lysis occurs, so which means the bacterial growth will not be there. And again, if you observe in the second method or second mechanism of action, they interact the nanoparticles in gram-negative bacteria. Thereby, they usually dephosphorylate the peptides of tyrosine residues. So once they dephosphorylate the peptides of tyrosine residues, certainly it's not going to good for the bacteria. Finally, it leads to the cell lysis. An interaction of AGNPs, which is silver nanoparticles with cell wall, and release more free radicals on bacteria. Finally, it leads to cell lysis. And this is another mechanism, but this is on uptake by endocytic vesicles. So to explain this, it will take much time. However, I will quickly go through it. So on mitochondria, it produces lots of reactive oxygen species, which means the free radicals. And through that, they do the oxidation of proteins, lipids, and DNA, finally leads to apoptosis. Same with nucleus, they arrest the cell cycle and antiproliferation, so finally apoptosis. And here you can observe cytoskeletal injuries, apoptosis, and macrophages. This also leads to inflammation. Here you can observe increase or elevated levels of reactive oxygen species, TNF and interleukin 6. And laser-assisted hypothermia, so they usually denature the nucleic acids, which is DNA and RNA, proteins and enzymes as well. So, in another way also, this leads to angiogenesis, and it inhibits that. So finally, starvation, apoptosis. So this is one of the articles which I have reported on nanotechnology and its applications in diabetes. Diabetes is the main core area of my research. So where I have given various information about nanotubes, artificial pancreas, nanopumps, microspheres, nanoshells, quantum dots, layer by layer methods, fullerenes, smart tattoos, all these things are really, very well equipped systems, and they are very good in detecting and also for the applications in the health systems and other areas. So this is the article which I have mentioned all those nanotubes, everything. In the next picture, you can observe the hypothetical picture, which you have nanosensors here, and then automatically whenever the blood glucose levels raised, automatically the sensors gives a beep, and it is just like tattoo. So it gives the beep, or it gives it continuously monitors your blood glucose levels. Once the blood glucose levels are raised, so they give you beep, or they give you the signals. Then if it is lower or if it is higher, then certainly the individual can take necessary actions. So that's how usually things are. But recently what I have read, even these are already in practice at some of the developed world. So even nanotechnology is being used in the diagnostics, drug delivery, and even tissue engineering, one of the most advanced technologies nowadays being used at the advanced laboratories. in the Western countries. So this is another example showing that nanotechnology being used in the neurological disorders as well. And this one, which we have clearly depicted in 2016, we have reported that the nanoparticles are clearly invading or clearly killing the cancerous cells. We have tested with a few cell lines, then they were showing very clear results, as anti-cancer activities. So this we have tested with some of the Malaysian medicinal plants, which shows a very good result. And coming into the research focus in my lab is basically whatever I do, it is completely linking with the human wellness. So natural product. So past decade, I'm completely working on natural product research of herbs. And my core area is diabetes, oxidative stress, antioxidants, cardiovascular diseases, and nanoparticle research. So these are the complete niche areas of my research, which happens at my laboratory, and also my students are working on various aspects. Finally, the conclusion will be into human wellness. So this is one of the anti-diabetic work we have published recently in 2017 with the honey. Usually people say honey is not good for diabetes, but there are various reports saying that even my research also shows that honey also good for diabetic patients. So biological and therapeutic effects of various types of honeys. This is another one which I have shown you the other article with the nanoparticles. So this is actually we have done the isolated compound for diabetes. So this one, which I was mentioning about, this clearly shows anti-cancer activity of Urochima longifolia branch extract mediated silver nanoparticles. So this is the one which I have given the 3 different mechanism of actions. This is on agriculture. This is on cancer. So this is again how the formation of nanoparticles occur, capping reactions. The functional groups work as a reductant, bioreductants. So they mingle with Ag ions, silver nitrate, and then they form as silver nanoparticles. Once we formulate the silver nanoparticles using plant materials, so then they can be easily accessible for various types of activities based on the phenolic compounds or based on the secondary metabolites are acting in the formation of nanoparticles. So here we can say that antibacterial, antioxidant, antifungal, antidiabetic, anti-tumor, based on the components they have, they can possess the biological activities. And finally, we know that nanotechnology will change the world. It's time to think about how. So this is actually from one of the scientists from MIT. So he has clearly mentioned that we know the nanotechnology will change the world. Because nanotechnology, as I have shown some of the slides, what areas or what fields they can show the applications. So which means nanotechnology is everywhere. In every field, nanotechnology is being used, and they are trying to use the nanotechnology for various types of applications, from small applications to larger applications, even in aeroplanes, and also solar systems, even buildings. Also, they are using nano sheets and nano materials or nanomaterial related products, which are with less weight and also long durability. And they, at the particle size, they usually possess large surface volume. That's why they can have more drug onto the surface when we inject into the body of an animal or also a human. So they can go into very narrow places also. If the site is very narrow, then even these smaller particles can reach to the places very easily. So conclusion is nanoparticles play an important role in the human health with ameliorating effects of various diseases such as diabetes, obesity, cancer, and neurodegenerative disorders. So not only this, even cardiovascular diseases, even other diseases as well, even lots of nanoparticles are being used for communicable diseases. Communicable diseases are like COVID, but not for COVID so far, but other communicable diseases or infectious diseases, we call them as. So the nanoparticles or nano medicines have been used or are being trying to use to ameliorate the effects of these infectious diseases and also non-communicable diseases. So, yeah, thank you. But yeah, I could see some of the questions here. Okay, question from one of the participants. Yeah, saying that what is the main mechanism of antimicrobial activity? Yeah, antimicrobial activity, which I have shown very clearly in this particular slide, the three mechanisms, if you observe in this particular slide. So this is the clear-cut picture which has given very hypothetically and which was also been reported by various other researchers also in the same manner. So I have summarized in this particular article, even we have published it in 2015. And so this particular article is clearly showing that the cell lysis happens in these three manners. Maybe, in other manners also, the bacteria or the microorganisms can be wiped off. And another question here is about anti-cancer activity. So is the anti-cancer activity same with all cell lines? Yes, the cell lines, whatever the activity we test on various types of cell lines, say for example, ovarian cell lines or cervical cell lines, cervical cancer cell lines, breast cancer cell lines, but it depends on the activity of the particular cells, particular nanoparticles. If the plant materials or the plant phytophenols have the capacity to inhibit those cancer cells, then only the nanoparticles can inhibit. But nanoparticles cannot just directly inhibit any kind of cancer cells based on the bioreductant available in that particular nanoparticles, then only it can show the better efficacy and potential activity. Yeah. And many others are just saying thank you. Yeah, thank you everyone. And my sincere thanks, humble thanks to Professor Sudarshanam sir for having me here in this international webinar on nanotechnology in current scenario. Thank you, sir.
Thank you, Dr. Visweswara, for explaining with good diagrams. I hope our audience will be definitely appreciating your presentation. As an alumni of Sri Venkateswara University, we are also thankful to you. Whenever we requested you for giving a talk, you have readily accepted and giving a nice presentation. Thank you very much.
Thank you, sir. Thank you, sir. My pleasure. Thank you, sir.